as the gold standard. Not all of this is stuff that I'm like an expert on. It's just stuff I remember from class, so it might not be the same as what your teacher says or like how someone else would explain it, but it's just meant for relaxation and enjoyment. So yeah, today's genetics video is going to be some more like basic biology information, so something I'm definitely not an expert on, but my last few videos have been a lot more in the weeds, so this one should be a little more general. We are going to be talking about the cell cycle and mitosis, meiosis, and then like some things that can go wrong with meiosis that lead to disorders like Down syndrome, for example. So yeah, I hope you enjoy. As always, we have my lovely drawings, so please ignore my drawing ability. I did my best. But the first thing we are going to be talking about is the cell cycle in general, and this is what the vast majority of the cells in your body are going through right now, all the time. So yeah, I'm actually going to start here, even though it is the last stage of the cell cycle, but it will help explain what's gonna happen in the cell cycle. So this is the M phase, which stands for mitosis, and that is where your cells actually divide, because mitosis is the division of your cells. It's like if you get a cut on your arm, your body has to regenerate those skin cells. It regenerates skin cells anyway all the time. That's why you lose dead skin, but it is the division of cells to make more new cells that are exactly like the old ones. So the last phase of the cell cycle is when the cells actually divide to make two new cells, and that's called mitosis, which we're going to talk about coming up in the next few minutes. But in order for that to happen at the end of the cell cycle, the first phase of the cell cycle is called the G1 phase. And I always imagine, I don't know if it's actually what it is, maybe it is, but I just imagine that G stands for growth in G1 and G2, because the main thing that happens is that the cell grows. Obviously, if you're going to make two cells from one cell, you gotta make it bigger so that it can make two like normal sized cells, so they have the right amount of stuff in them. So the cell has to grow in G1. G0, I know this looks like it just says go, but it's G0. It's like kind of a phase where cells enter into when they are not dividing. So it's drawn like outside of the cell cycle, because those are cells that aren't currently going to divide and go through the cell cycle, but they could enter back into the cell cycle at any time if we need them to continue to divide. But that's just a little thing that basically says not all of your cells are going through this all the time. But once a cell passes through G1, so it grows, everything's good, it goes into the S phase, which is, I guess it stands for synthesis, DNA synthesis, and that is where all of your DNA replicates. So every cell in your body has all of your DNA. It has 46 chromosomes for most people, and that DNA has to replicate. It has to make a full new copy of itself in order to divide, because each new cell needs all of the DNA. So you gotta replicate it first, then you can break the two cells apart and make two new cells with the right amount of DNA. So all of your DNA is going to replicate in the S phase. And then if that happens properly, you move into G2, which is essentially just more growth, getting ready to actually divide. So the cell grows more, and then it moves into the M cells will then make 4 cells because each of them would go through the cell cycle at mitosis. So that is the basics of the cell cycle. This is just saying that these 3 stages are all called interphase. So that's like everything that's not mitosis. So the cell cycle can be broken up into interphase and mitosis, or G1, S, G2, and M. So yeah. The only other thing is that I did write checkpoint here. That is basically just saying that for every stage of the cell cycle, or like at a lot of points in the cell cycle, there are checkpoints that essentially verify that everything is going properly. Because if your DNA is mutated, or if a cell is not growing properly, you don't want it to divide. You don't want it to make new cells that are all going to be like that are going to be mutated. You want to get rid of it or fix what's going wrong. So you have all of these checkpoints that basically make sure the cell is growing properly, make sure the DNA is replicated accurately, make sure it's growing properly, and then it's like, okay, now you can do mitosis. So the main way that that kind of comes into play is with cancer. Maybe you already know that cancer is essentially just mutated DNA, cells that have mutated DNA and are not normal cells, and they bypass the cell cycle. So like they just go through the cell cycle even though they're mutated, and that's what leads to like a big tumor that keeps growing, because it's these cells that have all these mutations. They're not good cells, and they just keep growing and growing and growing. And that can happen when the checkpoints are kind of like malfunctioning. And so you have what are called tumor suppressor genes that are involved in the checkpoints, and as the name suggests, they suppress tumors because they essentially make cells die when the DNA is not replicated properly. But if something is wrong with your tumor suppressors, like having a BRCA1 or 2 mutation, if you've heard of those, then those tumor suppressors are no longer working properly, and they're not assisting with these checkpoints properly, and so mutated cells go through the cell cycle and cause cancer. So that's how super high level overview of how like a BRCA1 mutation can lead to cancer is because it's not keeping the cell cycle in check, and so it just goes and goes and goes without being stopped. So that's the cell cycle. I'm gonna do just a brief overview of the S phase, which is the DNA synthesis or DNA replication. So all of your cells in your body have 46 chromosomes that look like this. So they are drawn 2 copies of each color, because that's 2 copies of each of your 23 chromosomes. You get 1 of each chromosome from your mom and 1 of each from your dad. So you have 2 of chromosome 2, chromosome 3, chromosome 4. So this is like, let's say these are chromosome 1, you got this one from your mom, this one from your dad, and then these are like chromosome 20, and right? Okay. So that is how most of your cells look. You have your 46 chromosomes, but during the S phase, before your cells divide, remember we have to replicate our DNA, and so it's going to look like this, where each of these chromosomes is going to replicate itself and create a whole new one, and the same chromosome, just different copies of it, right? So if you have 2 chromosome 2s, those are homologous chromosomes because they're the same one, right? And this is just a brief, like, diagram of what DNA replication kind of looks like. I'm not gonna go too deep into this with, like, the 5' and 3'. I won't talk about that. But what I will say is just that DNA replication is semi-conservative, which means that we have this original DNA strand. So, like, from here up, we'll say is this blue one, it's the original strand, and it is opened up, so unwound, kind of like a zipper, and then each of those 2 strands is copied. So each new strand, these are like this blue one and this blue one, is going to be one strand from the original DNA and one new strand. So that's what the meaning of semi-conservative is it's not like the old strand and then a new one. It's one of the old and then one new, makes the 2 new DNA strands. Like I said, I'm not really going to talk about 5' and 3' and Okazaki fragments. I could make like a whole video on DNA replication. But yeah, 5' to 3' it's just like the polarity, the direction of DNA, kind of like an arrow where, you know, you've got this end and you have the arrow end and it's gotta go in that direction, right? Like, it's got 2 different ends, and so that's like 5' down to 3' is the direction of a DNA strand. It kind of governs the way that DNA can be replicated, but if you want a whole video on DNA replication, I can do that. This is just to show that we're starting with this, we go to this, which is double the amount of DNA to prepare for mitosis. So, yeah. Alright. So now let's talk about everybody's favorite thing, which is mitosis. Alright. So, what I showed before with the duplicated DNA, that's what the cell will look like at the end of interphase, which is like, you know, G1, S, G2, but now we're going to the M phase. I will say that, like, there are so many different levels of detail and complicatedness with which you can explain mitosis. I'm going to give you a very low, like, basic level, but there are, I mean, if you want to break up these phases even more, there are, like, so many phases you could talk about. I'm giving you the basic, okay? Okay, so the first phase of mitosis is called prophase. Prophase. And in this phase, you've got your cell still, and you've got those 4 duplicated chromosomes, like I talked about earlier. They have not mutated colors. I just ran out of marker ink, so imagine they're still the same exact colors. Your eyes are not tricking you. And so what I didn't really mention before is that all of your DNA is in the nucleus of your cell, which is like another membrane inside of the cell membrane. I didn't draw it on the previous slide, but in prophase, basically the nucleus is going to start going away. So that's prophase. 4N just means that we've got 4 of each chromosome. You're supposed to have 2 of each chromosome in every cell. Because we duplicated them, we've got 4. So N is like the amount of genetic material, right? We've got 4N because we have 4 of each thing. Okay. So now we're moving on to metaphase. Metaphase. I always kind of remember metaphase because meta means like middle. And in metaphase, the chromosomes are lining up in the middle. So each chromosome, each of them is going to line up in the middle of the cell, and those centrioles are at opposite poles of the cell, and they have their microtubules that are coming in and attaching to each chromosome. So they're attaching to the middle of each of these chromosomes, and they're lined up at the middle. So we're getting ready to yank them apart and make 2 new cells. Okay, so we've got that, everything's prepared. Now we have anaphase. Anaphase is where they are actually coming apart. So these microtubules start to shorten and they are pulling the chromosomes towards the 2 poles of the cell. And so these are bent, kind of imagine it like someone's pulling you away and you're like, so that's why they're like bent, because they're being pulled by the middle of them. So they're pulled to each side. Each side is going to be the same. It gets 1/2 of the genetic material that was in there. And then we have telophase. And telophase is where it starts to, so the cell is actually starting to divide. Like I said, it's so complicated. Like there are so many different phases you could talk about, and these kind of run together. It's not like everything is mutually exclusive. Like late in anaphase, the cell does start to divide, and you'll see like a little bit of cleavage in the cell. Like it's starting to happen. It's not mutually exclusive. It's all kind of happening as one process, but I'm just trying to break them up simply. So the cells are starting to divide. The nuclear envelopes are reforming. We've got our chromosomes in each cell. And then in cytokinesis is when they actually divide. It's more complicated than that. There's a lot of process, but we now have 2 new cells. The chromosomes are in the nucleus, and we're back to 2N, which is the amount of DNA that humans have. You have 2N DNA, or 2 copies of each chromosome. So now, at the very end, it looks like it did before the S phase. 2 cells that are exactly like the original cell. So yeah, I don't know if I went through that really fast, but prophase, metaphase, anaphase, telophase, cytokinesis. That's how we get 2 cells from 1 cell. Okay. Yeah, I feel like I went through that fast, and like I said, mitosis is not like my specialty, so if anything is slightly inaccurate, I am sorry. Yeah, like, I know how to explain the process, but I don't know all of the words for everything, so, yeah. But now, we are going to talk about meiosis. I like meiosis more. It's definitely a lot more pertinent to genetics and genetic counseling because, like, meiosis is how disorders happen, like how you get chromosomal disorders and deletions and duplications and things, so, that's more related to, like, genetic counseling, which is what I do. So, let's talk about meiosis. So, what's happening is very similar to mitosis. The only difference, the main difference, is that this is only happening in your germ cells. So your germ cells are typically cells in the testes or the ovaries that are going to give rise to sperm and eggs. So those are what create babies when you do the deed. So, yeah, this is the process by which you create sperm and eggs, which is the process by which you make a baby. And so this is very important, because if it doesn't happen properly, you can't just like kill off the cell. Like it's supposed to create a baby. Whereas in mitosis, like if something happens incorrectly, the cell can just die, and it's fine if like one of your skin cells dies. So yeah, but we're creating a baby here. Okay, so we have a new cell, regular cell. This is a germ cell, we'll say, and the DNA has already replicated. So you can see there, like the X shape, we've got some replicated chromosomes. And I know this is kind of confusing based on how I drew it before, but for the purposes of this, these are homologous chromosomes. Even though I drew them in different colors, they are homologous chromosomes. So these are both chromosome 3 or whatever, this is the one from your dad and this is the one from your mom. And these are also the same chromosome. But for the purposes of meiosis, it's important that they're drawn in different colors. But yeah, I tried to draw them like different sizes so that you can see that like these are the same and these are the same. Does that make sense? Okay. So, one difference between mitosis and meiosis is that in meiosis, you're essentially going to go through all of those stages twice. So you have meiosis 1 and meiosis 2, and in each of those, you're going to have a prophase, a metaphase, an anaphase, a telophase, a cytokinesis. So in prophase 1, the first thing that happens, something is going to happen called crossing over. If you saw my genetics pop quiz Wednesday, I asked you, what phase of meiosis does crossing over occur? And the answer was prophase 1. So now you know, now you understand what I meant by that. And that is the process by which the homologous chromosomes exchange DNA. So they're actually going to cross over with each other at what's called chiasmata, and they're going to exchange some of their DNA. So I just drew it with one crossing over event, so you can see that like the blue and the pink exchanged a little piece, and the green and the orange exchanged a little piece as well. But this would normally happen like several times. So you'd have a whole mix of DNA, and that's basically just a way that like genetic variation happens. If your chromosomes didn't cross over, then essentially like there would be a lot of combinations, but there would not be like an infinite amount of babies that you could create, if that makes sense. Because like, I don't know, if your mom has these 2 chromosomes and she gives you like the whole one of this one, she could give your brother the whole one of that one too, and it's like you'd be so genetically similar to your brother. Does that make sense? So like with crossing over, it increases genetic variation, where you're getting this like jumbled mix of your parents' chromosomes rather than just like exactly one half. I don't know if that makes sense, but okay. So once crossing over has occurred, you can ignore this cell now. It's the original cell, but we've got this one that's going through meiosis, and as we know, it's 4N DNA, so 4 copies of everything, all of the genetic material. And the first thing that's going to happen is that the homologous this other cell got that one and that one. So now we have two new cells that are both 2N DNA, but they've got different homologous chromosomes. Yeah, I think that's all I wanted to say about that. I guess the one thing I will say, it's important to note that this is completely random. You have to imagine that there are 46 chromosomes in here, not 4. I'm just drawing 4 because I can't draw you 46 chromosomes in one cell. But there are 46, and it's just as possible, equally as possible, for this cell to get that one and this one. Like, it's, each segregation is completely random. It's not like you get the right half in one cell and the left half in one cell. You can get any of them, but it's always going to be one of each homologous chromosome. So one chromosome 1, one chromosome 2. So that's the end of meiosis 1. But now we're gonna keep going with meiosis 2. And so now it's going to be kind of like mitosis, where the sister chromatids are gonna separate rather than the homologous chromosomes. So the sister chromatids are gonna get pulled to each cell, and each one, again, is going to get one of each of your chromosomes. 1, 1, 1, 2, 1, 3. So here we've got these two. So that side of that one and that side of that one that didn't have any crossing over. And then we've got all the options. So it creates 4 different gametes. These are what are the eggs or sperm. That's what the eggs or sperm are going to look like. So they've got those chromosomes, and then so it's 1N. You have one copy of each of your chromosomes. And now when this combines, let's say this is an egg, and it combines with a sperm, they're going to fertilize, and you're going to get a zygote that has 2 of each of your chromosomes. And so that's how you get a baby that has the normal amount of human DNA, 2 of each of the 23 chromosomes, 46 chromosomes total. So yeah, whereas mitosis is going to create, 2N, it's dividing once, this is dividing twice, and we get 4 cells that are all 1N DNA, half of the DNA with 23 chromosomes. Yeah, so that's how you make a baby, everybody. Yeah, I think that's all I wanted to say about that. That's meiosis. So ideally in meiosis, each of the 4 daughter cells, each of the 4 sperm or egg, would get 1 of each chromosome, 23 chromosomes. When it combines with the other gamete, the sperm or egg, you get 46 chromosomes, 2 of each. Everything's normal. However, that does not always happen. Errors can happen in meiosis, and you can get abnormal gametes. So we are going to be talking about nondisjunction. So for the purposes of this, we are only looking at 1 chromosome. I know before we were looking at 2 different pairs of chromosomes, but now we're only looking at 1, just for simplicity. So imagine that this cell has the other 22 pairs of chromosomes. It's completely normal. And they are all going through meiosis normally. So the daughter cells are getting 1 of each of those chromosomes. But we are going to be looking at one of them, which is chromosome 21. You will find out why we are looking at chromosome 21. But these are homologous chromosomes, so they are both chromosome 21. Okay, so now we're going to go through has the other 22, but it's missing chromosome 21. So now, when these cells get fertilized by a normal sperm, this one is only going to have one chromosome 21. It's missing one, and it has one coming from the dad. So that is known as a monosomy when you have one of a particular chromosome. So this is monosomy 21, and that is lethal. It's not compatible with life. If this zygote forms, it's either going to not implant in the uterus at all, or it's going to be a very early miscarriage. It's not going to be a baby. There's never anyone been born with monosomy 21. But up here, when this cell with the two chromosome 21s combines with the sperm, now we get 3 chromosome 21s, 2 from mom, 1 from dad. And if you've ever heard Down syndrome called trisomy 21, that's what it means. Trisomy 3 of chromosome 21. So this is actually a scenario where the baby can be born. People with Down syndrome can live a very long, healthy life, albeit with challenges, but it is possible to live with trisomy 21, and that is how it happens, is through nondisjunction. I will say that, like other trisomies, so I'm specifically talking about trisomy 21, but this could happen with chromosome 1 or 2 or 5, but trisomy 1, 2, 5 is not compatible with life. It would be the same as the monosomy, where the baby would not become a baby. It would be a very early miscarriage, and that is because chromosome 21 is actually your smallest chromosome. The rest of them are bigger, and so having 3 of your smallest chromosome has the least effect, and so that's why you can be born with it. Whereas chromosome 1 is your biggest one. If you have 3 of it, that's a lot of extra genetic material that you're not supposed to have. So that can definitely, that's not going to lead to a normal baby. You can have trisomy 13 and trisomy 18. So 3 of the 13th or 18th chromosome, those are known as Patau and Edward syndrome. And they are, like, babies can be born with those. Oftentimes they're miscarriages, but they can be born. It's pretty rare for them to live past, like, a year old, because they're much more severe syndromes, but it can happen. So those are the trisomies that are compatible with life. The only monosomy that is compatible with life is called Turner syndrome, and that is where you have just one X chromosome. So I haven't really talked about X and Y chromosomes today, but this exact same process of meiosis happens with those sex chromosomes. So females have 2 X's, males have an X and a Y, and when they undergo meiosis, they're getting 1 copy of each chromosome, including the sex chromosomes. So all eggs will get 1 X chromosome, all sperm will get either an X or a Y, and then when it combines with an egg, that decides if it's a boy or a girl, if it gets XX or XY. Nondisjunction can also happen with the sex chromosomes, and so you can get, too many or too few sex chromosomes, and the only one where a monosomy is actually viable is where you have 1 X. So you're not getting a Y, you're not getting a second X, and that's called Turner syndrome. There are also other, like Klinefelter syndrome is where you get 2 X's and a Y, so it would look pretty much like this, just X's and Y. You can have triplo X, trisomy X. You can have XYY, I believe. trisomies and monosomies that are actually viable. The rest can totally happen, totally possible. It's just that you wouldn't actually see it because, you know, it's not viable, so it's not going to create an actual baby. The other thing I will say is that this non-disjunction can happen in meiosis 1 rather than meiosis 2. I'm just showing meiosis 2 to show you like all of these possible gametes, but it's totally possible for here, for this cell to look like that, and then for this one to be empty, because this one got both of those homologous chromosomes, and so then you'd get a lot of abnormal cells because this is empty, so both of these would have only one coming from the dad, and then both of these would end up being trisomies. So it totally can happen in meiosis 1 or meiosis 2. I'm just kind of showing you meiosis 2. The other thing I'm going to talk about, which is interesting, is why I have been saying that this is the mom and that these are eggs. That is because non-disjunction, and when you see like Down syndrome or a chromosomal abnormality, more often than not, that is coming from the mom. Totally possible for it to happen in the dad, in the sperm, but typically it is coming from the mom, and I'm going to talk about why that is. So I'm going to put this down for a second because my arm hurts, but the reason that happens is because females are born with all of the eggs they're ever going to have. You are born with all of your eggs even before you're born. You have them all. Whereas males, they're creating sperm all the time, right? If you know a man, you know he's creating sperm all the time. So like start to finish, you know, if he has a baby at 30 years old, like that sperm was fully created when he was 30 years old, like before the fertilization happened. Whereas for women, you have all the eggs before you're born, and they are arrested, like they are arrested means like stopped, not like handcuffs. They are just sitting in prophase 1. So like from the meiosis, we have all of our eggs, but they're just sitting like this for years and years and years in your ovaries from the time you're born. And they don't actually go through the rest of meiosis until you are ovulating that egg. So you've got all of the chromosomes already replicated, already sitting there, and they don't actually divide until you're getting ready to ovulate and create that baby. And so, like, what happens to everything in your body as you age? It kind of breaks down. It starts working less well. It's breaking down. And the same thing is happening to your eggs, to the chromosomes that are in your eggs. They're kind of breaking down, not working as properly. And so, essentially, as you age, as the female ages, the chromosomes, like the cohesins that are holding the chromosomes together, are kind of weakening. They're not as, young as they used to be. They're getting weaker, and so it's more likely for something like this to happen where they both go to one cell, they don't separate properly, or something like that, where something isn't happening properly just because those chromosomes are old. They've been sitting there forever. Whereas for a male, like if he's 60 years old, he just replicated those chromosomes. Even though he's 60, his sperm were just created. So that is why non-disjunction typically happens in the female, because her eggs are aging, whereas his sperm are not really aging. They're getting created all the time. So that is why chromosomal abnormalities typically come from the mom, and why the rate of them goes up chance of something like Down syndrome or trisomy 21. Yeah, so I don't remember the exact numbers, but like if you are a 25-year-old woman, I think the chance of Down syndrome is something like 1 in 1,000. If you are 45, the chance of having a baby with Down syndrome is like 1 in 15 or something, like very, very high. My mom was actually almost 43 when she had me, and there was like a 1 in 30 chance that I would have Down syndrome. So very high, because her eggs were old, her chromosomes were old. So not to scare ya, but that's how it happens. So that's chromosomal disorders. Next we're going to be talking about something else that I really like, and it kind of goes along with that. Yeah, but we're gonna talk about translocations. So nondisjunction, trisomy, monosomy are things that can happen that affect the entire chromosome. Like you have an extra or not enough, so it affects the number of chromosomes that you have. But there are a lot of other what are called structural rearrangements that can happen in your DNA, and they don't necessarily cause an issue for the person that has it, but they can cause issues for their offspring when their cells go through meiosis. So some examples of that I won't talk about are like deletions and duplications. Well, those would cause an issue in the parent, but like a little part of your chromosome can be deleted or duplicated. So that's an arrangement that would cause an issue because you'd be missing genetic material or have too much. But there are also inversions, so like a part of your chromosome can flip. So like let's say this bottom part flips, and so the material down here is up there and the material up there is down here. That in you, that's perfectly fine, like, because you still have all of the same genes, all of the same genetic material, so you'd be fine. It can cause issues during like crossing over because the chromosome isn't the way it's supposed to be. But the arrangement we're going to be talking about now is called translocations, which happens when genetic material from one chromosome essentially exchanges genetic material with another chromosome. So this one is called reciprocal translocations, and this can just happen like, usually it's like an accident, like your DNA breaks and then it just so happens to like combine with another chromosome. I don't really know like why it happens, but yeah, so these are 2 chromosomes, blue and gray. I did steal some stuff from my notes because this is a lot of chromosomes to draw and I didn't want to do that. So we have 2 chromosomes. Literally doesn't matter which ones they are. Let's say they're chromosome 11 and chromosome 22. Whatever. So something has happened where they broke. So both of them broke, and we get a normal blue one. So we've got like a normal 11, and then we have one that has most of chromosome 11, but a little piece of chromosome 22 on it, and then the same, a normal 22, meiosis, obviously we have to have our DNA replicated. So each of these is going to replicate. We have normal and then abnormal ones that are now replicated. And now they are going to go through meiosis. So the reason this is bad is because, like, this is treated as a chromosome 11, and this is treated as a chromosome 22, but they have bits that are not that correct chromosome. And as I said before, the segregation of the chromosomes is independent of one another. So, like, which chromosome 11 one cell gets has nothing to do with which chromosome 22 the other cell gets. So, like, if this is, like, the chromosome 11 that goes into one cell, and this is the chromosome 22 that also goes into that cell, well, now that cell is missing part of chromosome 11. It doesn't have this genetic material, and it has extra of this genetic material, extra chromosome 22. So that's what's bad, is when you're creating gametes. So this shows that. It shows all of the options that can happen for the segregation if you are a reciprocal translocation carrier. So over here, this is what's good. So what we can have is we can get a cell that has both of the untranslocated chromosomes. So this one and mirrored this one. So they're untranslocated. That's perfect. That's great. Now, when it separates again, we get normal chromosomes, and when it combines with the other gamete, the egg or sperm, we get 2 of each. We're perfectly fine. The baby doesn't even have the translocation. Now here, the cell can get both of the translocated chromosomes, so both of the ones that switch to DNA. So now here, the cell again is fine because it has the full chromosome 11 and the full chromosome 22. It's just that they're translocated. So, you know, it's okay, but yeah, it has the right amount of material. It's just translocated. So then, of course, we get cells that have those translocations, but still one of each chromosome. When it combines with the other gamete, we get 2 of each chromosome. It's just that the baby would have the same translocation as the parent, and so when that baby goes to have kids, they can have issues, but, like, for them, it's balanced. It's fine. Okay, now the bad stuff. So in these arrangements here, this is what I was talking about, where one cell gets, or like where they segregate independently, so you get a translocated and an untranslocated. So here we get the untranslocated blue one and the translocated gray one. This is hard because it's like mirrored. And then here you get the opposite. And so when you create the gametes, you can see that this one is missing part of the gray chromosome. It has extra blue, not enough gray. And then same over here, like, not enough blue, too much gray, because of the way that it segregated. And so now if you combine this with the other gamete, like here, you're going to get 2 normal blue ones, but you have this extra bit of blue, so that's called a partial trisomy. We talked about trisomy, like with trisomy 21. This would be a partial trisomy where you have an extra piece. It's not a full extra chromosome, but an extra piece, and depending on which chromosome we're talking about, that might be lethal. You might not be able to live with that, so that would be a miscarriage. Also, you would also have a zygotes that they create are abnormal and are going to be miscarriages, so they struggle to have kids. But it is possible, as you saw, they could have a completely normal child or one that carries the translocation. Okay, so that's reciprocal translocations. It can happen with any two chromosomes. They can exchange a little bit of genetic material, and you can get that. Lastly, last thing for today, we are going to talk about Robertsonian translocations. This is like my favorite thing. Obviously, I don't like them, but I like to talk about them. They're interesting. So yeah. Okay. So with reciprocal, you still have the same number of chromosomes. You still have 2 of each. It's just that some material swapped. With Robertsonian, it's a little bit different. So up until now, I have been drawing all of the chromosomes the same. I've been drawing all of the chromosomes kind of like equal on the top and the bottom. I guess I haven't even really talked about that the middle, this like dot, is the centromere, and then you have 2 arms. So like a top arm and a bottom arm. And I've been drawing them equal, so like the top is equal to the bottom. That's not actually how it is, and each chromosome is different. So chromosomes that are called acrocentric, they have a really, really, really tiny top arm, short arm. The short arm is really, really tiny, and all of the genetic material is in the bottom arm for acrocentric chromosomes. And the acrocentric ones are 13, 14, 15, 21, and 22. So those are the chromosomes that a Robertsonian translocation can happen for. So what happens here, I'm showing chromosome 14 and chromosome 21, which are both acrocentric, so they can create a Robertsonian translocation. And what happens is the exact same as a regular translocation, except you're getting the breakage of the chromosomes, and they are going to combine, the long arms are going to combine with each other, and you're going to get this. So this is this whole arm and this whole arm stacked on top of each other. So that is your Robertsonian translocation. The reason this can happen is because, or the reason it's like viable and doesn't cause any issues is all of the acrocentric chromosomes essentially have the same short arm. That little nub is not doing much. It's just like coding for ribosomal RNAs, but like if you don't have it, the other acrocentric chromosomes will take over. Like you don't really need that little nub. So those just get lost. They go away and you get this. But of course you're still going to have one normal one of each. So one normal 21, one normal 14. Yeah, you're still normal. It's just you have one that is translocated. So total, you've got 2 21s and 2 14s. It's just that one of your 14s and one of your 21s is combined. So these people actually only have 45 chromosomes, which would kind of look like a monosomy, but it's not. They're perfectly fine because they have the right amount of genetic material. But when they go through meiosis, these are all of the different ways that they can segregate. So these are the gametes that are made the different ways that these 3 can be separated. So you can get a cell that gets the normal 14 and the normal 21. All good. When it gets fertilized, you get 2 of each. Perfectly fine. You're normal. Here, you are only getting the translocated one. So you only get one chromosome, and it's the translocated one. And when that gets Turner syndrome is the only viable monosomy, so those are going to be lethal as well. But here, if we get the translocated one and then a normal 21, so the opposite of this one, now when it gets fertilized, we are going to get, so a normal one of each, right? And then like the normal one from the dad, and then here we have the translocation. So we have 2 blue ones, 2 14s, that's normal, and 3 orange, 3 21s. And so what is that? Trisomy 21, and we can get Down syndrome. So yeah, that's one other way that you can get Down syndrome. One is nondisjunction, the other is if the parent has a Robertsonian translocation. So that is, I believe, the only way that you can have like inherited Down syndrome. The vast majority of the time it happens through nondisjunction, which is like the parents didn't do anything. They are not carriers of Down syndrome. They're not like, they did not pass on Down syndrome. It's just a mistake that happened. With Robertsonian translocations, that's one way that you can actually sort of pass on Down syndrome. Like, it's because of the arrangement of chromosomes that you have that you passed on Down syndrome. So I think typically if you have a baby with Down syndrome, they would get like genetic testing to see if it's from a translocation or just from nondisjunction, and that kind of tells the parents like, okay, if it's from a translocation, you have a pretty high chance of having another baby with Down syndrome. But if it's not, then like your risk just goes back to what everybody else's risk is. Like, you're probably not going to have a baby with Down syndrome. So yeah, that is translocations, meiosis, mitosis, cell cycle, all that jazz. I hope you enjoyed that. I hope you have a new appreciation for how you came to be and the trials that you had to go through to be a normal human. Whenever I learn this stuff, I always am like, man, I'm surprised everything worked out the way it was supposed to when I was born. Like, crazy everything that can go wrong. But yeah, I hope you guys enjoyed this video. Thank you so much for watching. As always, I talk very fast. I'm very like hyper when I get to explain genetics, so sorry if it sucked, but this was fun. It took me a lot of work, so I really hope that you guys enjoyed it. But yeah, sleep well everyone,